So is your lab all clean for hazardous waste?
Huh?
Is all your hazardous waste clean?
You know, I'm in a new lab, so there's not much to worry about.
Well, that doesn't really say much because Dr. Snyder has stuff there from like the last people.
Well, that's exactly. So, and I have a new lab meeting.
Oh,
And
no.
the room that I'm in.
Oh, you're lucky.
Okay, guys. I know we're excited.
Super excited.
Super excited about the end of the semester. We're gonna have a short-ish lecture today. We're kind of done, and I'm using the rest of this time to introduce you to cool things you might not have learned about otherwise, and things that I have never, like, because we've been doing the exams and recitation, it's awesome because we haven't had to go super fast at the end, and we haven't been behind, and we're actually early, and everyone else, all the faculty are like, what? I'm like, yeah, that's me. Okay, so we're going to talk today about mobile genetic elements. I'm going to introduce you to them. And then Monday is going to be review / a little bit of more about how mobile genetic elements have been controlled. Because you need to control DNA that jumps around, otherwise that's not it. Okay, I saw a hand up. Yes?
Do we have a quiz this weekend?
No, there's not really much to have a quiz on. Yeah, no, we're done. We're done with everything, pretty much, except all the work is my work now, called grading your projects and writing stuff at that pesky final exam. Yes?
I do have a quick question. For recitation, we only have 7 quizzes.
For recitation, I keep getting this from people. You're dropping one.
Okay.
You're dropping one, and it's best 10 of the weekend quizzes. And please do not spend your time calculating your grades, spend your time studying for the final. If you are on the edge, please do not spend the time calculating your grade. Email me and ask me what your grade is. Because that's a waste of your time, right? And that's what people like to do, is they like to calculate their grade rather than study, because they think if they know they have a 78, then they're going to somehow get an A by calculating their grade rather than studying. Okay, so yes. Any other questions? We move on. Exams will be posted on Blackboard by end of day. I graded them all. I just need to do math and post. Math is hard. Okay, are we good? Yeah? Okay. Final May 20th. An expression project due this week. For those who haven't finished it yet, submit your proposal through SafeAssign. It is up. Sorry, I meant to delete that. It's up. Most people have submitted already. We're good. Okay, so for the next exam, these are the topics that you need to be focusing on. Binding of correct amino acids to tRNA, translation, initiation, elongation, termination, DNA damage, the types of damage, mechanisms of repair, and these are a bunch of them, errors from replication and how they are repaired, and then the double-strand break repair and DNA damage response. And then today we're going to do transposable elements. Finish that up on Monday. And then you guys can go, woo, party time. Semester's over. Yay. Okay, I'm happy. I could use a break and a half.
Yeah, we
Okay. Okay, so who's heard of mobile genetic elements and transposons before? Good. Who's never heard of them in their lives? Good. So I'm doing this for a reason. Great. So these are exactly what they sound like. Pieces of DNA, mobile genetic elements, pieces of DNA that move around in your genome. Okay? So some people call them jumping genes. They make up a huge percentage of our genomes and are highly responsible for genome evolution. About 50% of our genome contains kind of relics of these elements. Kind of amazing. And so this figure is just showing what our genomes look like. And so transposons make up about close to half. And then that's also in the repeated sequence categories because the ends end up being repeated as well as the actual transposon itself. And then what is this, 10, 20, 30% or so is this non-repetitive DNA that's that's not in introns or codons, and then genes are really only, what, 10 to 20%. I think I've seen 12% for coding genes, or for genes that make proteins. So it's pretty wild. So the thing to think about when you're thinking about, what does this have to do with genome evolution? When these elements jump around in your genome, they drag with them pieces of neighboring genes. And also, when they insert themselves in the DNA, depending on the type of element, when DNA polymerase goes to repair where it's been inserted, it ends up copying sequences also. So that's also how you get some duplications of sequences. Okay? So there's 3 classes of transposons. DNA-only transposons, which are exactly that. They never have an RNA intermediate. They're just DNA, and they just move around by hopping out of the DNA to another part of DNA. They encode their own transposase. That's the enzyme responsible for taking them out and putting them in. Really simple. What is one that you may have heard of? Well, P elements. You may have heard of P elements. Then there are the retroviral-like retrotransposons. You guys know you have retroviruses in you right now? Not really. Kind of. And these encode their own reverse transcriptase and transposase, also known as integrase. And they're called retroviral-like retrotransposon because they have this RT step, reverse transcriptase step, that's required. And we'll go through that. A common one is the TY1 yeast, if you may have heard of that. So they move via RNA, an RNA intermediate that's produced by the promoter that's in the transposon sequence itself. Then there's these non-retroviral retrotransposons, and they are slightly different. They also move via an RNA intermediate, but they move a little bit different. And so they encode their own reverse transcriptase and endonuclease, and that's how they move around. And we'll go through it. The most common one is L1. You know, you've heard of lines. How many people have heard, oh, our genome has all these lines and signs? L1 is a non-retroviral retrotransposon. That's one of the lines. Okay. So DNA-only transposons are largely responsible for the spread of antibiotic resistance in bacteria. You see them a lot in bacteria. Okay, and so this is just showing 3 different DNA-only transposons and how, along with the transposase gene that they all have, so there's their own transposase gene, so they bring their enzyme with them that is responsible for moving them around. They also bring with them resistance and resistance, type resistance genes. Okay, so there you have, so that's one. So how do they move around? They move through 2 mechanisms. One is just a cut and paste, and that's what we'll go through here. And another is replicative transposition. The only difference is in cut and paste, it's literally what it sounds like, where it's cut it out of where it is, paste it into somewhere else. With replicative transposition, it actually leaves behind a copy. So there's a replication step involved. So we'll just briefly go through the cut and paste mechanism. So you'll have your transposon in your DNA, and it encodes its own transposase. And that transposase is going to bind to these the inverted repeat sequences on either end, and then create this loop intermediate that's actually quite similar to what happens during the splicing reaction. And the actual transesterification, I forget, but quite similar. And so it pops out, so that leaves you with a broken chromosome, and then it goes into a new place. And notice your transposases are staggered here, and so that results in a staggered break. and it integrates in, and then the DNA polymerase comes and fills in what was broken because you've got the staggered break, and that's how you get these repeats that appear around transposons. So I was going to ask you guys, how is this break repaired here? We just learned about DNA repair, so I figured I'd make you guys integrate. Let's integrate. Come on, you guys are gonna graduate, you're gonna be bio majors. Remember, so what kind of DNA damage is that? Double strand break. So there were two major kinds: non-homologous end joining and a homologous recombination, right? And so, how does so do you think that repair through one of these mechanisms might result in more mutation? Non-homologous end joining, right? So sometimes you'll get repair, you can get restoration of the transposon if you use homologous recombination, because it's actually using the template from the sister chromatid to repair. If you're using non-homologous end joining, you lose your transposon because it's gone, and all you're doing is joining together your DNA, and then you actually increase mutation because, well, there's a break that's being repaired sloppily. Very cool, huh? Right. Any questions about this? Isn't it cool? Did you guys have any idea your DNA was so active? Do you think it was just hanging out in your cell doing nothing but expressing genes? It's crazy. I think it's awesome. So this is why I wanted to teach it to you guys, because I don't think I really learned about this until grad school. And the reason I learned about this is because the lab that I worked in was working on ApoBac3G, which a group in this, who's here and who's working on that project? Yeah, there you are. Where's the other one? She's gone. You're on that project. Anyways, ApoBac3G, they think may have evolved in a way to control activity of these genetic elements, which is kind of amazing, because if you have lots of DNA jumping around, I mean, it's good because you're increasing diversity, and if it's deleterious, you won't survive. But but still, if you have too much moving around, you can get, I don't know, I think more cancer for sure. So it could be bad. But so there's this whole class of proteins, the ApoBac proteins, that actually go around and hypermutate, and it's believed that hypermutation of these transposon sequences helps to inactivate them. And so if you compare humans to other species that have higher transposon activity, humans have way lower transposon activity, and we have had expansion of these ApoBac genes. I think it's like A through H, like a whole bunch of them. So it's kind of amazing. And so that's when I had to learn about this. And I was just like, whoa, it's kind of mind-blowing. So I'll talk a little bit about that part of it on Monday. Okay, so this is your DNA-only transposon. Replicative transposition, it's similar. That was cut and paste I just showed you. But the replicative transposition leaves behind a copy. Replicative. Okay, so some fun facts. Who's heard of VDJ recombination? Okay, who's taken immunology? Okay, so there's a process in maturation of T cell. Well, basically, generally, in creation of T cell receptors and antibodies, where you've got this shuffling of DNA and pasting together different sequences to create this diverse repertoire of antibodies and T cell receptors that we need to survive, right? You need highly diverse T cell receptors and antibodies, because if you've got if you have one antibody, you're going to be very successful at fighting infections. Right, you'll be highly successful at fighting one infection, but the rest of them, you're dead. T cell receptors too. So you need this huge diversity, and there's this whole process in creating that diversity that's called VDJ recombination. If you ever take immunology, you'll learn about it. Otherwise, you'll probably never learn about it. But this process of VDJ recombination is believed to have evolved from this cut and paste mechanism. And then also another fun fact is that viruses use this mechanism to integrate into host genomes. Another kind of cool thing. And so two examples are bacteriophage and retroviruses such as HIV. And so this is going to take me to HIV. So HIV encodes its own transposase. It's not called transposase, it's called integrase. And if you look at the life cycle of HIV, it gets into your cell, it's got an RNA genome, you have reverse transcription from RNA to DNA, then that DNA is integrated into your genome. Who thinks that there's integration of viral genomes into our genome all the time? How about, who thinks that it's every single virus that does this? Or who thinks it's just specific viruses? Okay, so it's viruses that have integrase. Those are what can integrate. So people actually, I'm just correcting what I've heard many times. People think that viruses integrate into genomes, and that's because when you take microbe, you're taught about bacteriophage, and you think you just kind of extrapolate. But if you don't have a mechanism for integrating into a genome, you're not going to. And actually, most viruses don't integrate into host genome at all. Yeah?
How do those viruses get us to replicate them?
How do viruses that don't integrate into... They have their genome, it's just existing, sitting next to ours. And if you're in the area where the appropriate enzymes were, you're going to replicate, right? You don't need to be integrated. Yeah. In virology, you'll probably learn all about this, but just wanted to correct that confusion that a lot of people have. So it has its own integrase. So there are these retroviral-like transposons, and they move in and out of chromosomes using that same mechanism. So they have an RNA intermediate that's then reverse transcribed into DNA and integrated in. Now, the only difference between a retroviral-like transposon and the HIV virus, what do you think the difference is? Right, HIV virus can kill you. The retroviral-like transposon, well, you're living with it right now. How come you're not spreading that around and infecting people and killing people? Like, what's the difference? Has anyone ever thought about this? Of course not. Let's think about it now. Right, so retroviral-like transposons, let's go back a minute to this chart and we can think about it here. So they have their genome, which basically contains LTRs, so long terminal repeats at each end, and they encode a reverse transcriptase and an integrase, and they move around that way. Okay, so what, do you think there's any way for this to get out of your cell? No, I'm seeing some people now that there's no way for it to get out of your cell. It's stuck in your genome, right? So HIV virus has a lot more genes, including capsid proteins, envelope proteins, all these proteins that package the virus into the genome into a virus that then could leave the cell. And so that's the difference there. But so these retroviral-like transposons, they contain the reverse transcriptase, so it's similar, and they have the integrase that can integrate in, but they can't actually ever get out. Yeah?
What if there was, say, fluid contact and a T cell were to attack this foreign cell, rupture it, and expose these elements to
So if there's a T cell from one person attacking a cell from another person,
And somehow the cells lysed and the DNA from the other person become exposed, and that exposure can then allow for some sort of integration into the new.
can the exposure from a T cell attacking another cell cause transmission of a retroelement? Well, no, only because when a T cell attacks another cell, it kills it in such a way that there's not ever going to be this exposure, and the other cell gets killed anyways.
Is there a way that a cell could be lysed inside another organism that scenario could happen?
Is there a way that a cell could be lysed inside of another organism where that scenario can happen? I'm not going to say no because I don't know everything, but I think it's highly unlikely in humans. I think when you start talking about bacteria, you do have frequent transmission of, a lot of these elements. The DNA only retrotransposons. But I don't think so. Yeah, I mean, nucleic acid on its own doesn't like to be out of its place for very long. It's usually rapidly degraded. There's nothing protecting it. When you have a virus, you have that nucleic acid packaged inside this beautiful protein coat and the membrane usually, which protects it, and that's what helps the transmission. So I'm going to say it's highly unlikely, but I've been wrong before. Okay. Any thoughts? Any questions? Okay. So then there's non-retroviral retrotransposons. So they're retrotransposons because retro usually means they have a reverse transcriptase. But these are non-retroviral retrotransposons. And they move using an endonuclease and a reverse transcriptase. And these are your lines. So you'll have it in your genome, and then you'll get expression of your L1 RNA. And then that will result in expression of the reverse transcriptase and endonuclease, which will bind to the L1 RNA. And it'll cleave the target strand, and then the target strand actually acts as a primer for the reverse transcription of itself, and then it's integrated into the new position in the genome. So that's a different way that they move around. Any questions? Oh, so that's where I stopped it for today. I guess on Monday, come excited to learn about how we learn how to control these in our genomes, and come with questions about things that you're worried about on the exam. Now, I mean, I don't want to hear that you're worried about your grade, but hopefully at this point, you've already told me you're worried about your grade. But if you have anything that as you begin to study, please make a list and bring it on Monday. Okay?